Hello everyone, in this video we'll be discussing prothrombin time and activated partial thromboplastin time. As mentioned in the previous discussions, secondary hemostasis refers to the cascade of enzymatic reactions which ultimately results in the conversion of fibrinogen to fibrin monomers. The following tests are classified under secondary hemostasis, which includes your PT and APTT. Secondary hemostasis involves coagulation and the formation of fibrin clot. Coagulation is a process whereon a vessel injury, plasma proteins, tissue factors, and calcium interact on the surface of platelets to form a stable fibrin clot. Platelets also interact with fibrin to form platelet-fibrin clot. Now, the coagulation cascade is a mechanism that consists of a series of cascading reactions involving development of enzymes from their precursors known as zymogens, which will further be converted to an activated state called serine protease. It has three pathways: the extrinsic, intrinsic, and common pathway. These pathways interact together to form a stable blood clot. The extrinsic and intrinsic coagulation pathways both lead into the final common pathway by activating factor 10. The extrinsic pathway begins when there is injury to the endothelial tissue, exposing factor 3 or the tissue factor to the blood. Once activated, it involves the initiation of tissue factor and its interaction with factor 7. Meanwhile, the intrinsic pathway begins when factor 12 or the Hageman factor is exposed to the collagen, kallikrein, and high molecular weight kininogen, and is subsequently activated. Factor 12A activates factor 11 into 11A. With the calcium ion, factor 11A activates factor 9. Then, factor 9A, factor 8A, and calcium form a complex to activate factor 10. The common pathway may result after the activation of factor 10 at the end of either pathway. The common pathway begins when factor 10A, 5A, and calcium bind together, forming a prothrombinase complex. The prothrombinase complex then activates prothrombin or factor 2 into thrombin factor 2A. Next, thrombin cleaves fibrinogen or factor 1 into fibrin or factor 1A, forming now your clot. Afterwards, thrombin cleaves the stabilizing factor into 13A. Factor 13A binds with calcium to then create fibrin crosslinks to stabilize the clot. For the specimen collection for both PT and APTT, it requires the collection of blood in plastic blue top tubes. This contains the anticoagulant of 3.2% sodium citrate. Prothrombin time is performed to detect coagulation factor deficiencies associated with the extrinsic and common pathway. This test is also used to monitor treatment with Coumadin or warfarin. To manually perform this test, we would need a venipuncture set to collect citrated blood, test tubes, water bath for incubation, pipettes for the aspiration and transfer of samples, and reagents and a stopwatch. The primary reagent used here is a tissue thromboplastin. And finally, we also need a control plasma, which is to be tested simultaneously with the sample to ensure the reliability of the entire test. To determine the prothrombin time, we need citrated plasma, which may be obtained through the centrifugation of citrated blood for 5 minutes. Afterwards, the plasma should immediately be separated from the red cells by transferring it to a clean test tube. This is then to be incubated together with the control plasma in the water bath for 2 to 3 minutes at 37 degrees Celsius. While waiting for the incubation step, you may prepare two separate test tubes, one labeled as test and the other labeled as C for control. Proceed to add 0.2 milliliters of the reagent into both of these tubes. When 3 minutes of incubation has elapsed, transfer 0.1 milliliters of the patient's plasma to the tube labeled T and 0.1 milliliters of the control plasma into the tube labeled C. Then, start the stopwatch. Mix both tubes and tilt them back and forth until clot formation is observed. Once this happens, stop the timer and record the time for both tubes. The result for the control plasma should be within the normal limit ranging from 10 to 13 seconds for the test to be considered valid. Take note that normal values may vary depending on the reagent used by the laboratory. In most cases, the prothrombin time is reported together with the International Normalized Ratio or INR for standardization of results. Next test you'll be performing is the activated partial thromboplastin time. This is a non-specific test of the intrinsic system pathway of coagulation. It may be prolonged in the presence of factor deficiencies, inhibitors, and other therapeutic inhibitors of coagulation such as heparin. It is prolonged in the presence of prekallikrein or high molecular weight kininogen deficiencies unless the test is performed using a reagent that contains ellagic acid as the activator. To perform this test, we would need a venipuncture set to collect citrated blood, test tubes, water bath for incubation, stopwatch, pipettes, APTT reagent plus calcium chloride as the reagents, and a control plasma. The calcium chloride, which will replace the calcium removed via chelation, is first incubated in the water bath for 5 minutes. To obtain the needed sample, the same centrifugation step is performed for APTT. The separated plasma is then transferred to the tube labeled T for test and the controlled plasma to the tube labeled C for control. 0.2 milliliters of the APTT reagent will then be added to both of these tubes, which are to be mixed thoroughly. These will then be incubated in the water bath for 5 minutes at 37 degrees Celsius. After 5 minutes, 0.2 milliliters of the pre-warmed calcium chloride will then be added to both tubes. The timer will be started immediately after. Both tubes, which are still in the water bath, should be tilted every 5 seconds and should be removed after 20 seconds. Continue tilting both tubes. Stop the timer when clot formation is observed, which normally occurs in less than 35 seconds.